Good morning, everybody. Welcome back again to the second day of the meeting. Thank you, everybody, for waking up early for this second hot topic. I remind you the hot topics were chosen by the committee out of all the abstracts that were sent for consideration, and we choose them because we thought they were work showing the latest and more relevant things for the patients. And today we will have 4 talks, all women. You go, girl. And we actually, I'm very happy to see that the 4 selected abstracts, they come from really all around the world. We have Nigeria, Israel, Belgium, and Australia represented today. So I think we cannot go better than that. Unfortunately, one of the presenters couldn't make it to Barcelona, so she has sent a video instead, and I hope that that will be still enjoyable. We will take all the talks first in a row, and we will leave the questioning for the end. There are, as you know, microphones on the sides that you can approach to ask the questions. We also have the app, so if you are too shy, you can write your question in the app and I will read it at the end. So with no delay, let's start with the first presentation, which is the Genome-wide association identifies novel etiological insights associated with Parkinson's disease in African and African admixed populations. The speaker is Oluwadamilola, that was difficult, Ojo, from Nigeria, and as I told you, this is actually in a video.
Thank you very much to the session chair, Dr. Romero Ramos, for the introduction. Good morning, everyone. I'm Oluwadamilola Ojo from Lagos, Nigeria, and I'd like to thank the organizers of the 6th World Parkinson Congress for accepting this abstract and allowing a video recording of this talk titled Genome-wide Association Studies Identifies Novel Here are my disclosures. Background, we know there's a gap in knowledge spanning the pathophysiology, clinical characteristics, and the genetics of Parkinson's disease in underrepresented populations. Despite advances in the field of genetics of complex diseases such as Parkinson's disease, populations of African ancestry remain understudied, especially when we think about population-based studies. Genome-wide association studies help scientists identify genes associated with either a particular trait or a disease, in this case, Parkinson's disease, by testing the genome of many individuals, looking for small variations known as SNPs, and identifying those SNPs that occur more frequently in the people with the disease, for example, Parkinson's disease, than in people without the disease. And though genome-wide association studies have been instrumental at identifying common variants in Parkinson's disease, unraveling the genetics of Parkinson's disease, this has largely been carried out in persons of European ancestry. Underrepresented populations such as Africans and persons of African ancestry are important because they are known to harbor the greatest genomic diversity worldwide. And these populations offer unique opportunities for studying genetics of both sporadic and monogenic forms of Parkinson's disease because they have the largest proportions of within population genetic variability in the world. The limitations in ancestral diversity in Parkinson's disease genetics is corroborated in this figure from this review paper by Sirugo et al., which shows that more than 75% of the information we have regarding genetics is based on European populations or persons of European ancestry. There are both social and scientific imperatives to understanding the genetic basis of a complex disease, such as Parkinson's disease, across global populations. And as medicine moves towards precision medicine and therapeutics, this will certainly have implications for the deployment and development of precision medicines, as we really cannot be precise until we understand the architecture of Parkinson's disease globally, and by globally meaning across all populations and ancestries. The Global Parkinson's Genetics Program, otherwise known as GP2, was created as part of the commitment to dramatically change the status quo and promote diverse representation in genetics of Parkinson's disease. And it's is focused on improving the understanding of the genetic framework of Parkinson's disease by creating a global research community working together to use the knowledge of Parkinson's disease genetics to accelerate the development and deployment of therapeutic strategies for PD, a disease for which a cure is the greatest unmet need, and to achieve the objective of inclusivity in PD genetic studies. GP2 and the Michael J. Fox Foundation funded the first studies extensively collecting data from Nigerians via the Nigerian Parkinson Disease Research Network that is part of IPDGC. In the last year, GP2 set up a collaborative project with IPDGC Africa to conduct the first PD-GWAS in persons of African ancestry using Africans, specifically Nigerians, African Americans, specifically from the Southwest USA, and then the African Caribbeans, the latter two groups herein referred to as the African admixed populations. To make this project possible, the Nigeria Parkinson Disease Research Network teamed up with Black PD standing for Black and African American Connections to Parkinson's Disease, GP2, 23andMe, to conduct this collaborative study. The team is composed of 24 Nigerian institutions, 4 Black PD sites at the time of sample collection, and a total of 78 authors, 46 from African institutions. This schematic displays the general workflow of the study. And once the data had been split according to ancestry, we first conducted the GWAS on the African population, then the African admixed population, followed by a meta-analysis. Thereafter followed a number of follow-up analyses, including haplotype analysis, short and long read sequencing, and homozygosity patterns. Here we see some demographic and clinical characteristics of the cohorts. Data consists of individual level data from Nigeria, and then the GP2 release 5, and then summary statistics from 23 and me. The GWAS meta-analysis strikingly revealed a novel African ancestry-specific genome-wide significant signal associated with Parkinson's disease risk at the GBA1 locus. This displays the top SNP, which is an intronic variant. And contrary to what we would expect when assessing common variations linked to Parkinson's disease risk, a high odds ratio was identified for this signal. It was initially thought that this SNP could be tagging a coding or a structural variant to explain the association and giving the observed large effect size of the signal. However, we did not find a coding or structural variation to explain this signal. Our study indicates that individuals carrying the risk allele, which is RS3115534, have a 1.58 increase in PD risk compared to those without the risk allele. There was low heterogeneity in the effect estimates between the African and African admixed individuals, supporting the finding as a robust one. Figures from the 1,000 genome study, which is not shown on the slide, show that this variant is very rare in persons of European and Asian ancestry, and slightly present in admixed American populations, and that's because they obviously harbor certain levels of African ancestry. When looking at African subpopulations as shown on this slide, we see that the variant is more frequent in Nigerian populations than other African populations. And this actually shows the Nigerian founder effect of this haplotype, where we see an almost 30% frequency for the risk allele in the Esan of Nigeria, versus, for example, 17% frequency in Webuye in Kenya, which is in East Africa. An interesting point is how different the genetic structure of PD, Parkinson's disease, in persons of African ancestry is when compared to Parkinson's disease in persons of European ancestry. Here we randomly sampled a similar number of Parkinson's disease cases, 1,200, and controls, 2,445 for both populations and at overlapping SNPs. And this figure, combining the two GWASs, reveals the huge effect size of the GBA signal in the African and African admixed population compared with the European population. We also assessed whether this GBA1 variant is associated with the age at onset in Parkinson's disease, and linear regression analysis showed that the risk allele was also a modifier for the age at onset, resulting in Parkinson's disease onset 2 years earlier per risk allele. Genomic admixture versus linear regression analysis also revealed that the risk allele was positively correlated with the percentage of African ancestry, again confirming an African founder effect. In conclusion, our study has identified a novel ancestry-specific genetic risk factor. Because it is an intronic variant and not a structural or coding variant, as most GBA variants are known to be, this finding points to GBA1 regulation as a major mechanistic basis for Parkinson's disease in persons of African ancestry, and gene expression can be explored as a novel therapeutic target for clinical trials in this population. This finding highlights the importance of understanding population-specific genetic risk in complex diseases and places a spotlight on an entirely new population for precision therapeutic clinical trials, which were hitherto reserved for a subset of persons of European descent. We would like to thank everyone involved on this project for fantastic teamwork. Thanks to all our collaborators and investigators, Nigerian Parkinson Disease Research Network, IPDGC Africa, Black PD, GP2, and 23andMe. We'd like to thank our funders, especially the Michael J. Fox Foundation. And last but not the least, thanks to all our patients. And I thank you for your attention.